Bismillah, Assalamualaikum warahmatullah. You're listening to microbiology lesson on microscopy, and this is part 4, the last part of this topic. And in this episode, I'm going to briefly mention some basic techniques, especially staining techniques. We often use these for experiments related to light microscopy. One of the first techniques you learn when you step into a microbiology lab is the hanging drop preparation. What you need for this, firstly, is a concave slide. So that's a glass slide with an inward curve, like a crater. But what you do with this slide is you put a ring of petroleum jelly around that inward curve. It will make sense in a minute why we put that ring. The second step is you put that slide aside. Then you pick up a glass cover slip. You put a drop of your bacteria sample or protozoa sample, whatever, in the middle of that slip. The third step is you pick up the slide again, you turn it upside down. Then you put the slide on top of the glass slip. When doing this, make sure the sample drop is inside the concaved area. The petroleum jelly you put before will help the glass slip to stick to the slide. Then after that, you can simply turn the slide back upside again, and then it's ready for you to observe under your microscope. It's very simple. And you notice that we don't do any treatment towards the sample itself. We take it fresh from the pond or from wherever you get them, and you put them under the microscope. Because of that, the hanging drop preparation is exceedingly useful for observing microbial motility, or how microorganisms swim around. So that's your hanging drop preparation. The second technique is the smear preparation. So this one requires a bit more practice. It's not hard, it's just rarely the students get this done perfectly the first time. For this, you're going to need 2 slides. The slides we use for this one would be just normal slides without any concavity. What you do is, firstly, you drop your sample near the end of your first slide. Put a nice loopful there. Then you take a second slide. You put the edge of it on the middle of the first slide. You angle it at around 40 degrees. Then slowly drag it closer and closer to the sample drop. You'll notice when the second slide touches the drop, the fluid will spread out across the edge of your second slide. And when this happens, you simply drag the slide back to the opposite direction, all the way to the other end of your first slide. So if done right, you're going to have a smear, a thin film of sample. It's usually thicker on one side, the side of the initial drop, and it's gradually thinner as you go to the opposite end of your slide. Another way to make a smear is you drop your stain. We'll talk about stains in a minute. You drop a tiny bit of your staining solution on your slide in the middle. Then you use your inoculation loop to mix your bacteria with it and spread it out in a circular fashion. Unlike the one I mentioned just now, you don't need to spread it all over the slides. You just make it the size of a penny. Then typically we would heat fix the slide. You heat the slide by passing the slide on top of your burner a couple of times. That'll make the sample sticks to your slide. And then you can observe them. They won't move now, your bacteria, because the heat fixing pretty much killed them right away. Okay, so that's the hanging drop preparation and smear preparation. Now let's talk about those staining solutions. Many microorganisms that you will work with, you can't distinguish them from their background because they are colorless. If you recall from the previous episodes, you need strong enough contrast between your sample and its background to make the sample visible. One way to get that contrast is by staining your samples. You put colors on them. So a stain is a large molecule. It's made up of three components. The first component is benzene, that carbon ring molecule that you learn in your chemistry class. So the benzene itself has no color. The color of a stain comes from the second component called the chromophore. So them two, the benzene and the chromophore, when they're together, they are called chromogen. Remember that, yeah? Benzene plus chromophore are called chromogen. If you only have these two components alone though, the stain is not going to stick to your samples. For that, you need a third component called oxochrome. Oxochrome conveys that ionization property to your chromogen. In other words, oxochrome helps your chromogen to bind to the cells. It does this by making your stain either cationic or anionic. Now, what does that mean? When you ionize a stain molecule, the chromogen can have a positive charge or a negative charge. When the chromogen has a positive charge, we call them cationic chromogen. For example, if you have a stain called methylene blue, it can ionize into a large chromogen structure and a small chlorine ion. So the chlorine ion is negatively charged and the large chromogen will be positively charged. Because the chromogen is positively charged, we call it cationic chromogen. Now, why does this matter? Well, for one, it helps you distinguish between basic stains and acidic stains. Because basic stains have cationic chromogens. The acidic stains is the opposite. They have anionic chromogen, and anionic chromogens are negatively charged. Secondly, it also helps you understand why basic stains bind more easily to certain components of cells. For example, nucleic acids are negatively charged, right? So if I were to ask you, which of the two types of stain can bind easily to nucleic acids, the basic or the acidic? So you think about it. Well, nucleic acids are negatively charged, so it needs to bind to a positively charged chromogen. So positively charged chromogen are cationic. Cationic chromogen is a property of a basic stains. So therefore, basic stains will have an easier time binding to nucleic acids. See, you just need to memorize that basic stains are positively charged because it has a cationic chromogen. Alright, now that you have understood the principle, the rest of these are just straightforward and we can go through them quickly. So, staining techniques, you can categorize them into three: the simple stains, the differential stains, and the special stains. The first one is the simple stains. In simple staining, you use one dye, for example, just methylene blue or just crystal violet. It's not going to give you a detailed diagnosis, for example, if you use it to look for, if you use it to look at samples from infected patients. But simple staining is pretty quick and you can use it to detect the presence of foreign microorganisms in your sample right away. You can also use them to see the shapes of the bacteria. Are they cocci, which are spherical, or are they bacilli, which are, cylindrical, looks like a cylinder or a rod. You can look at their arrangements. Are they in clusters, the staphylo arrangements, or are they in chain arrangements like the short diplo or a long strepto? So yeah, that's where simple stains come in handy. The second category is the differential stains. For this, you use more than one dye, and you use it very often to differentiate between types of bacteria that are present in the same sample. Two common examples are gram stain and acid-fast stain. With gram staining, your aim is to differentiate between gram-positive and gram-negative bacteria. You want to know this to, for instance, to narrow down which bacteria cause your patient to get sick. So the gram-positive will be purple and the gram-negative will be pink. Now, the first thing is you get yourself a smear preparation. Then you flood it with a dye called crystal violet. This crystal violet is your primary stain, the first stain that will color all your bacteria, and all of them are going to be purple. Then you wash the slide gently, just with water, tap water. Then you add iodine. Iodine acts as a mordant. A mordant makes the primary stain binds stronger to the cell. In this case, our mordant creates a CVI complex, a crystal violet iodine complex. So it's going to make the color turns to dark purple. And then you wash your slide again. Then you put a decolorizing agent. In gram staining, we use ethyl alcohol as our decolorizing agent. For gram-negative bacteria, when you apply the decolorizing agent, their membrane will become more porous. They'll have more holes in them. And because of those holes, your purple stain will leave the gram-negative bacteria easily. And then you wash the slide again with water. At this point, your gram-positive bacteria still retain that purple color. Your gram-negative bacteria, on the other hand, will be colorless because of the decolorizing agent we just applied. And finally, to color those colorless bacteria, we flood the slides with a counter stain called safranin. So in the end, you'll get gram-positive bacteria, which will be stained purple, and then your gram-negative bacteria, which will be pink. In the microbiology lab classes, we usually give students Staph aureus for gram-positive and E. coli for gram-negative. And then we ask them to do gram staining. Now, why does this work? The difference between them is in the membrane structure. Gram-positive bacteria has a thick peptidoglycan layer, which is made up of like a lattice structure of sugar and amino acids. So when the cell absorbs the purple stain, the crystal violet, this thick layer stops that purple stain from leaving the cells. On the other hand, gram-negative bacteria also have peptidoglycan layers, but it's much thinner, like 10 times thinner. Instead, for wall reinforcement, they have an extra outer membrane made up of lipids. This lipid outer membrane gets dissolved when we apply the alcohol, the decolorizing agent. So the membrane becomes porous and the purple dye leaves the cell when we wash them. So that's how gram staining works. Another example of differential stains is acid-fast stain. We use it to differentiate Mycobacterium usually, to differentiate them from other non-acid-fast bacteria. Before I forget, the word fast here, it doesn't mean quick. I've seen second language students and even lecturers mistranslate this. The word fast here means tight or attached firmly. Like when you screw a bolt or something, you want to make it fast, you want to make it tight. So acid fast here means that once mycobacteria get the primary stain inside them, they will hold the stain tightly inside. They won't let the primary stain go away, even if you decolorize it using acidic agents. So the bacteria that can do this, we call them acid-fast bacteria. What you do with this staining is you flood your slide with a stain called carbofuchsin, and then you heat it up. You can use flame, but usually we use water steam to heat it up. You boil water under the slide to get the steam. That heat helps this primary stain to get into the cell. There is the Kim-Yoon variation of this method where you don't use heat, but don't worry about that for now. Just focus on this. You heat it up for like 5 minutes and then you let it cool down a bit. And then you wash the slide with tap water gently. After that, you apply the decolorizing agent, acid alcohol, not just alcohol like in gram staining. The agent we use here has a bit of hydrochloric acid. You drop it on the slides, drop by drop by drop. That will get rid of the carbofuchsin in non-acid fast bacteria. On the other hand, the acid fast bacteria like Mycobacterium will hold that carbofuchsin inside. Then you wash the slide with water. And after that, you flood the slide with the counter stain. Why? To color the non-acid fast bacteria. We use methylene blue as the counter stain. And you leave it for a couple of minutes, then wash it again with water, and you're done. And when you put it under the microscope, you're going to see the acid fast bacteria as red and the non-acid fast bacteria as blue. Yeah, that's it. The last category is the special stains. By the way, the categorization that I use in this lesson, don't get obsessive compulsive with them. Okay, they are just loose categories that some scientists use, and others may categorize these staining techniques differently. All right, so special stains help us identify the presence of special structures for various reasons. For example, negative stains work on microorganisms that have gelatinous capsules. We want to know that because the presence of capsules can make bacteria more virulent. Another example is spore stains, which works for staining the microorganisms when they are in spore stage. So it's when you want to see whether your bacteria do have that defensive spore structure, which may explain why they persist, why you can't kill them with your whatever current treatment. So how do we do them? For negative stain, the technique is simple. You don't even need to heat fix your sample. You put a loopful of nigrosin, mix it with your sample, and you do the smear preparation that I mentioned before. You put your nigrosin plus sample near the end of your first slide. You take a second slide, put the edge of it at the middle of the first slide. Then you angle it at 45 degrees. Actually, maybe lower, about 30 degrees or 40 degrees. Then drag it towards the sample. When the slide touches the sample fluid will spread along the edge of your second slide. And then you drag it towards the opposite end, spreading the sample all over the first slide. When you put your negative staining slide under the microscope, you're going to see that the cells themselves are not stained, but the area surrounding them is. This happens because in negative staining, we usually use acidic stains. Acidic stains, if you remember, have a negatively charged chromogen. When the negatively charged chromogen comes into contact with the negatively charged bacterial surface, they repel, they push each other away. And that creates contrast between the cells and its background. So negative stain as a principle, you can also use it even in electron microscopy. You're just going to use a different set of staining reagents, of course. But the idea is the same. In negative staining, instead of coloring the cells, you color its adjacent surrounding. All right, this is the last one, spore stain. We can use it to differentiate between endospore and vegetative structures of a microorganism. The primary stain that we use is called malachite green. We use it because it's one of the few that can penetrate the spore membrane. So we flood the slide with malachite green and we heat it up. That's going to enhance the penetration of malachite green into the spore structure. Once the malachite green is inside the spores, it will stay in there. But the malachite green won't stay inside the vegetative structures. And that's good because we now can differentiate the spore structure from the vegetative structure. What we do then is we decolorize the vegetative structure simply by using water. The water will wash off the green stain from the vegetative structures, but not the spore cells. Then we color the vegetative structures by counter staining them with safranin, which makes them red in color. In the end, what you'll see under the microscope are cells that look red, and they're going to have these green dots in them. So those green dots are their endospores. So that's how you do the spore stain. All right, that's it for this topic. Now, obviously, there are other staining techniques in all the categories that I mentioned in this lesson. You're going to learn them in other courses, especially your lab courses. But for this microscopy topic, you just want to get a good grasp on the scientific principles behind the staining techniques, and then maybe recognize a couple of examples from each technical category. If you can do that, you'll be fine. Okay, see you in the next topic. Barakallahu li wa lakum. Wassalamu alaikum wa rahmatullah.